name is Ian Jacobs and I am the Vice Chancellor of UNSW Sydney, and like so many of you, a specialist in ovarian cancer treatment and research. I also have the enormous privilege of being a board member of Ovarian Cancer Australia, and in that capacity it's an honour to give the introduction to this meeting. I'd like to start by acknowledging the traditional custodians of the various lands on which we are on today and pay my respects to elders past, present and emerging. It is a great honour to be here this evening, to be a part of this important symposium. And this year, Ovarian Cancer Australia is proud to present a compelling programme. It features international and Australian experts presenting the latest developments and advances in what is, I think we all agree, a rapidly evolving field of ovarian cancer research and treatment. It includes targeted therapy, PARP inhibitors. And due to the unpredictable situation with COVID-19, our advisory committee made the wise decision earlier this year to hold the event virtually over 4 sessions, 2 evenings and 2 mornings. I'd like to thank a wonderful lineup of guest speakers for giving their time to be with us today. And my thanks also to the advisory group who were instrumental in pulling together today's program. Professor David Botel, Professor Anna DeFazio, Dr. Dil Garced, and Dr. Tarek Menawi. Ovarian cancer, as many of you will know, is a national organisation founded in 2001 by people who wanted to provide support to women affected by ovarian cancer and their families and friends. Ovarian Cancer Australia's vision is to save lives and make sure that no woman with ovarian cancer is without support. We're confident that the generous sharing of ideas and expertise made possible by holding events such as this one will help ovarian cancer to achieve this vision. OCA offer a range of support services for women and their families, and that includes a support services helpline, telephone and face-to-face support groups, consumer forums, webinars, and our online forum. In addition, OCA's Teal Support Programme is a free national telehealth outreach programme which tailors support to each individual. It provides specialist ovarian cancer nurses, a psychosocial counsellor, and a social worker support opportunity for women throughout their diagnosis, their treatment, and beyond that. The resources of Ovarian Cancer Australia, which provide information on diagnosis, treatment, well-being, and support services, are all free of charge, and they can be achieved by accessing the Ovarian Cancer Australia website. I need to also thank AstraZeneca for providing the funding to enable OCA to hold this symposium, and a special thank you to the Ovarian Cancer Australia support team for their hard work and their dedication in bringing this event to life. Thanks so much to all of you. Now, without further delay, we can begin session 1 of the 2021 Ovarian Cancer Symposium on Genomics. And I'm delighted to invite the chair of this session, Professor Linda Mileshkin, who is Deputy Director of Medical Oncology at the Peter MacCallum Cancer Centre. Linda, thanks so much for chairing this session. I'm going to hand over the microphone to you. Thank you.
Thank you very much, Ian, for the kind introduction and for the opportunity to chair this session this evening. We've got quite a full program with some very interesting speakers. I just wanted to remind the participants that you can submit questions during the talks using the Q&A button at the right upper side of your screen. And if other people have posted a question you also want answered, you can click on the side of it on the up arrow to put that question up the list. So we might get straight into it. Our first speaker is coming to us from the UK. So we're delighted to have him with us. So Professor Ahmed is a professor of gynecological oncology at the University of Oxford and a consultant gynecological oncologist at the Oxford Cancer and Haematology Centre. He originally did his training in Egypt and then did his PhD and postdoctoral training in both Cambridge and at the MD Anderson. And we're delighted that today he's going to talk to us about the Oxford Classic and the story of the Spark cluster. So, welcome, Professor Ahmed.
Thank you very much for the introduction and giving me this opportunity. It's really great pleasure to be here, talk to you all. The collaboration with Australian ovarian cancer researchers have had a really big impact on my research interests and my career to date. So as I'm going to explain later in the talk. So thank you very much for the kind invitation. So just a quick disclosure that I'm a founder, shareholder, consultant, director of Singular Bio Limited. So this is the background is St. Hugh's College in the spring. This is my college in Oxford. This is the Weatherall Institute of Molecular Medicine, where my lab is based, and this is the Churchill Hospital, where the gynae oncology services takes place. I don't really need to tell this audience that ovarian cancer is about over 300,000 women diagnosed with ovarian cancer every year, with a 5-year survival of around 40%. More than 70% of patients diagnosed at advanced stage where cure is difficult to achieve, with a high-grade ovarian cancer is the most common type, and it's the high-grade ovarian cancer that my research has focused over the past many years. So my lab has really been very much interested in understanding the molecular drivers of clinical outcome in ovarian cancer. And for that, we chose what I really believe is a very provocative clinical entity, which is minimal residual disease, in order to study molecular drivers of ovarian cancer. I want to explain to you what I mean by minimal residual disease, as exemplified by this patient whom I had the pleasure of looking after, a patient at presentation. She had ascites, an abdominal mass, and a pelvic mass. And following neoadjuvant chemotherapy, this ascites disappeared, the mass disappeared both in the abdomen and the pelvis. And at the time of surgery, really we couldn't see any remnant of the tumor, with a CA-125 marker going from over 2,000 down to normal, below 30 international units per milliliter. When you look at the momentum and look at it by H&E, you find this minimal residual disease that cannot see by eye. It comprises about less than 1% of the tumor load, but for this particular patient, it meant that she came back with recurrence within 10 months of completing first-line treatment, which is really very, very provocative as to why and how that happens, in spite of excellent response to treatment and excellent surgery earlier on. So really we're focused on trying to understand the molecular drivers of minimal residual disease. So we've known for many years from this collaboration that I had the pleasure to work with our Australian colleagues, David Botel and Anna DeFazio, which was published a few years back now, showing that high-grade serous ovarian cancer P53 mutations are ubiquitous. And as we know, just really as a little background, I'm sure we will hear more about this later in the coming sessions, but as we know, the high-grade serous ovarian cancer is characterized by profound chromosome numeric aberrations as compared to low-grade serous ovarian cancer. And this is really just a demonstration of that. If you look at the variant allele fractions across the entire genome, these are a whole genome sequence sample obtained from a patient with high-grade serous ovarian cancer. This is really typical appearance with areas of loss of heterozygosity, deletions, chromosome amplifications involving either single arms or even single entire chromosomes across the entire genome. So this is like the key backgrounds of high-grade genomic characteristics of high-grade serous ovarian cancer, P53 mutation, and chromosome numeric aberrations. So I wanted to understand the mechanisms that drive minimal residual disease, and we started by trying to hypothesize, trying to address this hypothesis where a single cell becomes transformed into a cancer cell, and as these tumors progress, they accumulate more mutations and more genomic alteration. And the hypothesis was that then the chemotherapy provides a bottleneck that allows only the fittest and allows only the fittest clones to survive and then expand and go on to form recurrences. We were set out to test this idea, but we thought that if we really wanted to test this idea, if we take this tumor at presentation and a tumor at recurrence and look and compare the different clones here as exemplified by different colors of cells, one by looking at this may start to think immediately that these purple cells have certain genomic characteristics that give them fitness properties that allow them to resist chemotherapy and expand later on. However, we really thought about this that without really looking at minimal residual disease itself, one can't really make that conclusion, because if you look at minimal residual disease in patients with exceptional responders following chemotherapy and find that the purple-looking cells have particularly resisted the tumor and have survived, and then later on expanded, then that gives you the potential conclusion that these genomic alterations in this purple cells are actually drivers of resistance. However, you could also have a situation where you have equal proportional killing of all the different types of cells, but then later on expansion of these purple cells, and this would be either stochastic or the acquisition of mutations that are, post chemotherapy resistance, that are not in themselves driver of why these cells resisted chemodynamics. So really, to differentiate between these two possibilities, we designed the Oxford Ovarian Cancer Predict Chemotherapy Response Trial, or OXO-PCR trial, where we were taking biopsies pre-chemotherapy from patients who were treated by neoadjuvant chemotherapy. Then 3 cycles later, we go back, look at the same sites where we have obtained the biopsies from, because we mark those sites by surgical clips, obtain further biopsies, and then also take biopsies at recurrence opportunistically if those patients go on to have surgery. This is a video just to show you how we did this study. So this is pre-chemotherapy. You have peritoneal or mental disease. We take biopsies from those. This is also diaphragmatic peritoneum. We take biopsies, leave surgical clips, come back 10 weeks later after chemotherapy. You see surgical clip is there marking the site of biopsy, but the tumor had really completely disappeared. but these surgical clips act as a marker for us to sample those individual sites. We then subject those to whole genome sequencing. This was a really very difficult exercise because we started by trying various whole genome sequencing techniques. Some of them were published, like this complete genomics solution for this, which is the long fragment read technologies for sequencing a very small number of cells. But we quickly realized that we needed to really develop our own method of whole genome sequencing and analysis. And I'm not going to really go through the details, but we published this method about last year, which we call DigiPico, and a neural network analysis algorithm called MutalX that allows us to get very highly accurate whole genome sequencing analysis and accurate identification of single nucleotide variants from a very small amount of material. The difference, the problem when you attempt to do whole genome sequencing from such limited number of cells is that you really get profound false positive, profound number of false positive mutations that make downstream analysis extremely difficult. So we overcome this by this method that we developed and published last year. So the analysis from this was a little bit disappointing because it showed that this idea of a bottleneck and survival of the fittest wasn't really there. So if you look at this particular patient as an example, where you have, this is pre-chemotherapy, this is minimal residual disease, and this is the tumor and recurrence. And the size of these circles, this kind of represents a diagram of an evolutionary tree with the size of the circle representing the percentage of that particular clone. And if you look particularly here at this clone, you see that prior to chemotherapy, it represented about 5% of the tumor populations. And minimal residual disease still was 5%, but actually in recurrence, you see that it expanded. But that really indicates that that reason for expansion is really nothing to do with chemotherapy resistance per se at this stage, but it probably happened through neutral evolution or other mechanisms. So whether genomic alterations were driving minimal residual disease molecularly wasn't really, we were not able to prove that, at least in our hands, with this study designed directly in patients, obtaining materials directly from patients. We were not able to find evidence for that. So we started thinking about phenotypic plasticity and phenotypic variation, and we started looking at expression profiles of these tumors. And let me just give you the idea of that. It's the idea again. It's can we identify molecular? Can we identify molecular phenotypes, molecular-driven phenotypes that come with certain enhanced characteristics of those cells that drive their persistence and resistance to chemotherapy. And what we thought about was it was going to be very difficult to look directly into tumor cells. That's learning from what we know about high-grade serous ovarian cancer. As I mentioned, the disease of chromosome numeric aberration. And this experiment really exemplifies this. So if you look at single-cell sequences of normal and some cancer cells, these essentially the heat map represents gene expression across the different chromosomes from 1 to chromosome 22. And as you could see here, that this chromosome 6, where there's an amplification, and chromosome 22, where there's a deletion, drive the expression of that entire arm of the chromosome, making what we believe makes direct going into expression profiling directly to tumors extremely difficult to us, because you'll be unable to disentangle the effect of expression from the effect of the underlying chromosome numeric aberration. We wanted to understand expression differences between tumors and between cells as drivers of molecular of phenotypes independent from their underlying chromosome numeric aberrations. So to do this, we borrowed some concepts from other tumor types like breast cancer. So in very simple terms, we know that in breast cancer, there is a luminal type, for example, and a basal type of breast cancer. And if you look at their molecular characteristics, they're very much similar to the luminal type and basal type of normal mammary epithelium. So we thought, could we do something similar for ovarian cancer? And we asked the question whether we typified the normal cells of the fallopian tube as the origin of great majority of high-grade serous ovarian cancers. If we identify the subtypes of fallopian tubes, can we then get a good classification of the tumor that is independent from chromosome numeric aberrations, because we'll be looking directly into normal fallopian tube cells. Indeed, this is what we did. So we used a SmartSeq tool and looked and did deep sequencing of about 6,000 fallopian tube cells from patients either with cancer or no cancer. These fallopian tube cells did not have cancer, and we were able to identify 4 different subtypes in the non-ciliated cells, in the secretory cells, which we call differentiated, keratin 17, EMT, and cell cycle. We were able to validate that those were real subtypes, because when we look at organoids grown from single cells of the fallopian tubes, we can find that this is, for example, the keratin 17 cluster, which we believe differentiated from that single cell that gave rise to the organoid. So it made sense that this was a real phenotypic subclass of the cells in the fallopian tubes. We then went on to identify the markers that gave us the best separation between the different subtypes, and those markers turned out to be 52 marker genes. And the story of the SPARK cluster is that one of the key 52 marker genes in the EMT cluster was in fact SPARK. So I remember Z1 was my PhD student who did this work. And she came to me and I asked what are the marker genes. She said it's SPARK. I said, okay, so this is David Botel's SPARK cluster that he told me about when I was doing my PhD, and he was visiting Cambridge at the time as a visiting professor on a sabbatical. Kept telling me about the importance of the SPARK cluster. So when Z1 mentioned this to me, I started to take this very seriously indeed. So the first thing we did was we went back to this really iconic data set from the Australian ovarian cancer study by Tothel et al. And from David's lab, and we applied these 52 marker genes into the patients from this group, and we were able to reclassify these into the different components, 4 different subtypes of secretory cells, and in addition to the ciliated cells subtype, which actually turned out to be the low-grade tumors. But very interestingly, when we look at the correlation between the EMT scores of tumors and overall survival of patients, we found a strong correlation with poor survival, which is something that, again, David and others like the TCGA have reported previously. But the difficulty in previous reports that this classification of previous classification of EMT high tumors correlated in some data sets with survival, but not in others. But the reason I was really excited about this work is that when we looked at any published data set that had about 100 patients or more with expression profiles, be it on microarrays or RNA sequencing, we found a consistent correlation between the Oxford, what we call the Oxford class classification of ovarian cancer, the Oxford Classic defined EMT high tumors and survival. So that was very reassuring. And this is what C1, who's the student who did most of this work, has portrayed here in this diagram, that actually the normal cell phenotypes are mirrored into cancer. We then went on and took the 52 genes and designed a nanostring panel to undertake further validation collaboration with Christina Fotopoulou at Hammersmith. And reassuringly, we were able again to find that about 17 to 20% of the tumors from her cohort were EMT classified as EMT high, and that this was an independently correlated with poor survival, independent of stage, grade, residual disease, or in fact BRCA1 or BRCA2 status. So what I believe we have found with this is that it is something that David and other researchers have previously reported about this correlation between EMT and poor survival. However, I think we've refined the classification, because when we look, for example, at the TCGA data, we found that 50 of the patients were, according to our classification, misclassified as non-EMT. We reclassify them into EMT high using this classification, and this really resulted in this robust association with survival. But we're going back to the question about minimal residual disease. Is EMT high phenotype enriched in minimal residual disease? So we looked at the samples from the Oxford PCR study. So on the x-axis here, we have the post-chemotherapy samples, and on the y-axis, we measured the EMT score. And very interestingly, those samples that had the highest EMT scores were in fact coming from patients who had exceptional response and coming from minimal residual disease. So this paper was just published. We're grateful to JCI Insight for giving us the cover image. But really what we show is that these minimal residual diseases characterized by are essentially EMT high tumors, but also very interestingly have very strong lipid signature, in that they very much resemble adipocytes. And I just want to say they are not adipocytes, because we've spent a lot of time making sure that these are really cancer cells and not the neighboring adipocytes. And these cancer cells have a gene expression signature that is very much characteristic of adipocytes, both for fatty acid oxidation and, in fact, fatty acid synthesis. In fact, when we try to mimic this in the lab in vitro in different cancer cell lines, we find that these minimal residual diseases, this is the seahorse experiment looking at dependence of these cells on fatty acid oxidation. We found that these cells are really rely on fatty acid oxidation for their metabolism. So just this is the portrait of showing that these disguised cancer cells in minimal residual disease as fat cells. So what we believe is that minimal residual disease is a really highly relevant model to study mechanisms of cancer cell survival, and that the EMT high cell state is a strong indicator for cancer cell survival and poor clinical outcome. And how EMT high state drives cell survival is likely determined by complex interactions with the microenvironment, but also this very inherent characteristic in these cells of relying on fatty acid oxidation is something that is definitely worth noting, because it can open door for many therapeutic interventions. I want to end the talk by thanking C1, who's the first author who is responsible for this work on Oxford Classic, and Mara on her work on minimal residual disease expression profile, our collaborators, particularly Chris Yao and others, and our funders, particularly Ovarian Cancer Action in the UK. Thank you very much for listening, and happy to take any questions.
Thank you very much, Professor Ahmed. Just looking at the one question that's appeared. Kylie Gringe saying, fascinating talk. Do the different cell types in the fallopian tube have specific normal functions?
We have so far really focused on doing expression profiles and getting the molecular phenotypes. We have not really done any functional studies, but it is very interesting to see, for instance, that the keratin 17 subtype seems to have very interesting characteristics like expression of HLA type 2 molecules. And a subproportion of this appears to be also Wnt active, which really opens the door for whether a proportion of this particular subtype could work as a potential tissue-resident stem cell. We're looking into that and doing the functional work, but it's still really early days to pinpoint a particular function for these cells.
Wondering, what are you thinking that you do next? Any ideas how to target these adipocyte-like cells that are left at completion of chemotherapy?
So what we have explained in the paper that was just published, in fact, last week, is that those cells, if you block fatty acid oxidation in them, they really die. So if you take cells pre-chemotherapy and treat them with atomoxir, which is a fatty acid oxidation inhibitor, it doesn't have any effect. But if you look at the cells following chemotherapy and you treat those cells with atomoxir, you get a significant and profound cell killing. Atomoxir can't be used in clinical practice because it's very toxic. So, however, there are other fatty acid oxidation inhibitors that we are looking at. The other interesting point about these EMT high tumors is that they seem to be surrounded by macrophages of the M2 subtype. So we believe that there is a strong immunosuppressive effect on these and the environment of these EMT high minimal residual disease tumors that also could be targeted by immunological approaches.
Interesting. We've got another question from David Botel. It says, could EMT high cells survive due to a physical barrier or matricellome reducing chemotherapy exposure?
Potentially, David. I'm not sure. I think we can't exclude that, but I actually think that given that these cells have very strong differences and phenotypic differences, perhaps we could think about those phenotypic differences as drivers of survival. But we shouldn't be excluding the area of exclusion, the idea of exclusion completely of the list of possibilities.
Got another question. Lots of questions. You talked about that, we didn't seem to be selecting specific clones with the chemotherapy, but he wanted to know, have you explored emergence of clones of BRCA reverted mutations in early disease in germline BRCA mutation carriers?
Yes, so I think this is a very, very important question. So I think we have two perhaps different and complementary mechanisms of resistance. So in patients who have or tumors that have, let's say, an Achilles heel that is targeted by the therapeutic you're giving, in this case, BRCA1 or BRCA2 mutation that is targeted by a PARP inhibitor, or in some cases carboplatin, those cells could undertake mechanisms of resistance that are essentially like that reversion of mutations in order to avoid that Achilles heel. However, if in a tumor that doesn't have that such very very strong definer, genomic definer that you are targeting with your drug of choice or with your targeted therapy, the mechanisms are, I don't think that the genomic mechanisms, at least from what we have seen from analysis of patient samples, play an important role in resistance in the context of minimal residual disease following chemotherapy. Sorry, I couldn't hear what you said, Lynn.
One more question from Greg, who wants to know, are EMT high cells more likely in the primary tumour or in metastatic sites? And are there any specific anatomic sites likely to have a greater concentration of EMT high cells?
So we really didn't address this question directly, but what I can say is that from looking at multi-site sampling, because this is what we did for the Oxford Classic, for the OXO PCR study, we did multi-site sampling. For each patient, we took from 3 to 6 samples, including primary or metastatic site. What was really fascinating is that that when we classified something as EMT, a patient's tumor as EMT high, it tended to be consistent among the different sites, or at least more so than when you compared one patient to another patient. It's very interestingly that when we classify a patient's tumor as EMT high, we tend to see that it's EMT high in most of the sites, including the primary site.
Remains to thank you very much for your excellent talk and your wonderful work. And I might introduce the next speaker now.
Thank you very much. Thanks.
Thank you, Professor Ahmed. So our next speaker is well known to many on the line. It's Associate Professor Kylie Gringe, who's a group leader at Peter MacCallum Cancer Centre. She originally trained in New Zealand and did her PhD at Cambridge and then postdoctoral positions at Peter Mac and the James Cook University. And she holds a number of fellowships now where her research is focused on developing molecular prognostic models for pre-invasive breast lesions such as DCIS, but of specific relevance tonight she's had a large amount of focus on mucinous ovarian cancer, one of the rare and difficult to treat subtypes of ovarian cancer, and she's going to tell us a bit about some of her work in that space. So welcome, Kylie.
Good evening, everyone, and welcome to my seminar, which I'll be talking about mucinous ovarian cancer, and thanks very much for joining us this evening. So, as you all know, mucinous ovarian tumours are a very distinct subtype of ovarian cancer. They have different epidemiological factors, and we know that they don't respond well to conventional ovarian chemotherapies. So, when they're diagnosed at an advanced stage, the outcome's very poor. So you can see this in this Kaplan-Meier plot here. We've got the stage 3 and 4 MOCs, which do really badly. If you do catch it early, though, the stage 1 and 2s do quite well. And I'm also showing here this group, these metastatic non-ovarian mucinous tumours with these extra-ovarian metastases, which are a group that were historically often confused with primary mucinous ovarian tumours, to the point where it wasn't really even clear whether there was such a thing as a primary mucinous ovarian tumour. And so we were really interested in trying to understand the landscape of these tumours and the differences between them and the extra-ovarian metastases. So with my co-investigators, we kick-started this gamut study, and we had a few questions. So one of them was whether these primary mucinous ovarian carcinomas existed and how they developed. So we know that there are benign and borderline tumours that arise at the ovary and do very well, but it wasn't entirely clear whether they were the origin of the invasive tumours, particularly the high-grade invasive tumours, which are the most difficult to distinguish from metastases from other sites. We wanted to understand cell of origin and, of course, find targets for therapy. So this was a big study over many years, and it had 2 different postdocs working on it over time. We collected tumours from all of these fantastic collaborators, both here in Australia and overseas, and generated a large volume of genomic and transcriptomic data. And I'm going to just summarise all this work, which was published a few years ago in this slide. So in studying benign tumours, we identified that they mostly had 1, maybe 2 changes. And these were generally a KRAS mutation, a BRAF, or a CDKN2A event. And CDKN2A is the gene that encodes P16. From benign to borderline, they were very closely related. The borderline usually had 1 or more additional changes. So if it started out with a KRAS, it might pick up a P16 deletion, for example. But when you went from borderline to carcinoma, then you really saw quite a few things happening. So the biggest and most frequent change was the acquisition of a P53 mutation. And the carcinomas also had more large-scale copy number events, so gains and losses of regions of the genome. ERB2 was also more commonly amplified in the carcinomas and the borderline tumours. And the difference between the low grade and the high grade was that the high grade had many more copy number changes and the low grade was still fairly genomically stable. But we could still see a clear relationship between high grade MOC and borderline. And importantly, the changes that we saw were different from those tumours that you see and the metastases, and we were able to distinguish them from tumours from the colon, from the stomach, for example, or from the endometrium. The hardest one was actually the pancreatic tumours, which were quite similar, but we were very careful with our pathology, and I think with expert pathology, which is getting really good now to be able to distinguish these tumours, you can distinguish most of the primary MOC from those extra ovarian metastases. So to summarise that, we do know that primary MOC exists. We've kind of resolved this controversy with the extra ovarian metastases. We know that they can arise from borderline precursors, and they're distinct from these other tumours, but somewhat similar to pancreatic. So they've got the KRAS, P53 and P16 events, which is similar to pancreatic carcinoma. But what are the clinical implications, and what do these driving events, particularly new ones that I have identified, do? So I'm showing you here the kind of mutational heat map, if you like, of these mucinous tumours from benign all the way through to the grade 3s, and each of these columns is an individual patient sample. And you can see that other than the most obvious candidates, there were a lot of genes that were in that kind of 5 to 10% mutation rate. But one particularly caught our eye, which is this gene ALK3. Now, the reason that it caught our eye was because many of the mutations were truncating. So you can see on this lollipop plot, this is the protein structure of ALK3 with the 2 key domains, and the mutations are spread out across, but the black ones are the truncating, and they were the most common. So 8 out of the 15 mutations that we saw were truncating. And the missense mutations were all clustered in this ETS DNA binding domain. So ALK3 is a transcription factor, and this is the bit that binds to the DNA. So we hypothesized that ALK3 could be a tumour suppressor gene. And Carolina, who's a PhD student in the lab just about to submit, used CRISPR to knock out ALK3 in some mucinous ovarian cancer cell lines that we had in the lab here, and also in this immortalized ovarian surface epithelium cell line. And you can see the effect of this knockout at the DNA, RNA, and to a greater or lesser extent at the protein level as well. And this had phenotypic consequences. So on the left here is showing the impact on proliferation as measured by cell titer glow. And you've got the wild type in the pink, and in the blue is the knockout. And you can see in the 3 cell lines that we're seeing an increase in the proliferation rate, although actually the opposite in our hose line. They also saw differences in invasion. So in this invasion assay here, again, for the 3 ovarian carcinoma cell lines, here this is the wild type in the black and the knockout in the grey, we're seeing an increase in the invasion capacity of these cell lines, although no change for the surface epithelial cell line. So we don't really know the mechanism behind this, but we do know that it's mutated in these tumours with a high percentage of inactivating mutations, and that a knockout gives us pro-tumour phenotypes, but only in the carcinoma cell lines and not in that ovarian epithelium. So we also think that maybe there need to be other mutations present, first of all, that it cooperates with to further enhance tumorigenesis, rather than being an initiating event. So the next step is to repeat the knockout for some independent validation, including in additional models. To use RNA-seq to interrogate the pathways. It's a transcription factor, so we would hope to see the impact of that on the RNA at the RNA level. And also to potentially explore a haploinsufficiency model. So one thing that was interesting about those mutations was that they were mostly heterozygous, even though they were inactivating. So it seems to be a different pattern from that other tumour suppressor, P53. Which segues quite nicely into this the study of P53 that we did with Martin, who's a wonderful pathologist in Canada. And Martin developed an immunohistochemical screening method to be able to look at P53 in the mucinous tumours and infer what type of mutation that they had. So you can see these 4 different patterns here corresponding to the 4 main types of mutation. This was a slightly adapted method specific for mucinous tumours, as opposed to the one that was previously developed for high-grade serous. And so we were able to score a large cohort of carcinomas and also borderline tumours, and the overall frequency was similar to what we would expect. But what we were particularly interested in was seeing whether it was related to outcome. And it wasn't in the carcinomas, but it was in the borderline tumours. So here we've got in the sort of pinky orange colour, whatever that is, you can see the normal, as it had a normal P53 staining pattern or no mutation, and in the blue it had an abnormal, and you can see this survival difference in terms of progression-free survival and even overall survival, and that was statistically significant. So we think this could potentially be a biomarker. You know, these borderline tumours, they're really big, it's hard to sample exactly, but if you saw a P53 pattern in there, that would raise some warning flags. You wouldn't give them chemotherapy because we wouldn't do that for many of the early stage tumours anyway, but maybe an enhanced follow-up, and that's something that could be tested in the future. So coming on to treating the carcinoma. So again, here's this dire outcome for the stage 3 and 4s. And this is data from our cohort, which, keeping in mind, is quite old. And so the majority of women diagnosed at stage 3-4 received platinum-based therapies because that was the standard of care at the time, and still is for most ovarian cancers. The picture changed when they become recurrent. So these are the carcinomas that were either stage 3-4 and then progressed, or were non-responsive to their first treatment, or they were women who they might not have had treatment first of all, but they've had a recurrence and have been getting their therapies. And so for those women, they perhaps more likely to get a platinum as their first, effectively first line. But you can see the real diversity of therapies that are offered by clinicians to women at recurrence. And this really reflects the fact that there's very little evidence to guide a choice of chemotherapy, and that's a big problem. So, so why are we worried about them, so many of them still being treated with platinum? So we know that platinum is more effective in tumours that have a compromised DNA repair, and particularly homologous recombination deficiency. That's why the high-grade serous responds generally quite well initially. So we measured this genomic scar of using HRD, this HRD score for large-scale chromosome changes. And here I'm showing you the results from benign, borderline, grade 1 through to grade 3 MOCs. And for comparison, this is the BRCA deficient median from this paper here by Kelly and colleagues in 2016. And we can see from this that actually we only have one tumour that could be considered to be bona fide HRD deficient, like a few that were in the kind of, maybe not. But the vast majority have high genomic stability and would be very unlikely to respond to platinum and PARP inhibitors. So we can see that this genomic data tallies really well with the clinical data that these tumours are quite resistant to platinum and would almost certainly be resistant to PARP inhibitors as well. So what about a different sort of DNA repair deficiency? So historically, mismatch repair deficiency was thought to be one of the features of mucinous ovarian cancer, but actually this is probably due to those older cohorts being, contaminated with extra-ovarian metastases from the endometrium or from the colon that do have a reasonably high level of mismatch repair. So we can see in this graph, this is the tumour mutation burden, that we only had one out of over 180 MOCs that had a high mutation burden and a mutation in a mismatch repair gene, as opposed to 4 tumours that had one or the other of those in the extra-ovarian metastases. So on this biomarker alone, which is one of the biomarkers used to determine whether you can offer immune checkpoint inhibitors, they might not be effective for the majority of MOC. But this is a relatively understudied area, and we certainly could do with better biomarkers in case there are some that could respond. So targeted therapy is obviously where we want to go. These are the main driving mutations, and many of these, of course, have therapies either already in the clinic, such as those targeting ERB2 amplification, and those that are in clinical trials. And there are many more than just the few that I'm listing here. But how do we get evidence that they work in MOC? We know there are tissue-specific responses to targeted therapies, such as for BRAF. So will they work? And we really don't know. So we started out by testing these therapies using the existing cell lines that we had, and this was again work done by Carolina during her PhD. But rather than doing this in 2D, she developed a model to look at this in 3D, so using that matrigel as sort of a slightly more physiological way. And this was done at the Victorian Centre for Functional Genomics at Peter Mac. She tested a small panel of targeted agents in the existing cell lines based on the observations, mutations that we observed in those. And the results were fairly mixed, it would be fair to say. So, for example, I had high hopes for this RAS-RAF inhibitor, and the one KRAS cell line sort of had a response, but it certainly wasn't that different to the others. The palbociclib really didn't seem to correlate with the CDKN2A deletions. We had a bit of a better response, more specific to the mutations for this PI3 kinase inhibitor, but was really disappointed to see that this Residual inhibitor targeting the Wnt pathway really wasn't effective at all. And the APR246 targeting the P53 missense did have some response, and particularly in this cell line here, although this was P53 wild type, so we're not quite sure why it responded so well. But the problem, of course, with this analysis, although it's a good start, is that we've only got these few cell lines, and actually it turned out this OMC3 we don't think is mucinous ovarian primary at all. So it's not really enough to be able to draw conclusions around the mutation and whether they respond. So Carolina established this tumour organoid program for the lab, followed up now by Olivia, who's just started her PhD, where we collect the tumours at surgery and then put them through this processing to try and develop these organoid cultures and other parts of the tumour we collect, for example, for genomics analysis or histology. And this took a lot of time and a lot of effort from Carolina to get this up and running, but is now working extremely. This is the types of tumours that we tried, and our success rate since Carolina worked out what the best culturing conditions were has been over 80%. So we're really happy with how this is going. So the challenges were obviously finding the right growth factor cocktail, and it turns out that Wnt is particularly critical. So you can see in this images here, these are some organoids, ones that are growing in the 100% concentration, but 100% of the concentration that we found to be suitable, and even reducing it by half was enough after 3 or 4 passages that the organoids are very, very unhappy. So this is definitely a critical growth factor for these tumours. Other challenges, we're getting enough tissue, particularly from the carcinomas and particularly from the recurrent carcinomas. We're often working with really tiny amounts. And also an incorrect diagnosis. It's really difficult when someone's going in for surgery to know exactly what they have. So we ended up with a lot of benign and a lot of borderline tumours, and about a quarter of the ones that we got were actually turned out not to be mucinous ovarian after all. So this reduces our efficiency, and it's really frustrating. You get something in, it starts to grow really well, and then you get the path report back that it's not mucinous, so it's no good for us. So I'll just show you, this was the first carcinoma that Carolina grew successfully, ORG16, and you can see these organoids growing here, the histology that matched really nicely between the tumour and the organoid, the genomics that also match really nicely between the tumour and organoid. And you can see this is a tumour that has it's a bit of a classic P53 mutation, ERB2 amplification, and also a PIK3CA mutation. So of course, Carolina tried to do some drug screening, and you can see here we do get a bit of a response to this APR246, the P53 targeting agent, but really disappointing response to trastuzumab, and also lopatamab as well didn't work. Perhaps this is because of the PIK3CA mutation, but what I think we'll plan to do in the future is not just look at single agents, but start looking at at combinations of things. Maybe these two together could work really well for this organoid. So that's ongoing, but in the meantime, we also want to be identifying new targets, not just ones that we already know exist, but ones that might be more specific for mucinous ovarian cancer. This is a collaboration with Jessica Hollian at RMIT, and she's what I guess you might call a structural bioinformaticist. So she can take our gene expression and genomic data, and she maps it to the protein networks and also looks at the structure. So she's looking for those proteins that are not only highly expressed in our tumours compared to normal tissue, but that are sort of hubs for those protein networks and also are structurally compatible with designing drugs too. We can validate those in vitro, and then Jess again works the magic with doing in silico screens of millions of compounds to try and investigate leading compounds that then can feed back to us in the lab to do our drug screening and ultimately in vivo drug testing. So at the moment in this project, we're up to this bit here, this in vitro target validation. And Olivia, as part of her master's, did some knockdown using siRNA, again in our mucinous ovarian cancer cell lines, to try and identify those targets that would reduce proliferation in our cancer cell lines, but not so much in our ovarian surface epithelial cell lines. So that's what we're showing here, is the viability reduction compared to the hose. And we can see we've got some good targets here. You know, some of these are really obvious targets that we know are potentially targetable in other cancer types, but also some new ones that we'll be excited to explore, validating in our organoid models, perhaps using CRISPR and getting some small molecules to test as well. So we feel like we've got some exciting leads going forward. So to summarise, for mucinous ovarian cancer, I think we can understand why they don't respond to platinum and that PARP inhibitors are unlikely to be of use. There are several targeted therapy options, but it's difficult to work out which ones are going to work. You know, we can use the organoids and that will give us some clue, and it's definitely something we're going to do in the future. But clinical trials that are specific for mucinous ovarian cancer are probably going to be impossible to conduct outside of the context of a basket trial. So we should definitely be advocating for MOC to be included in basket trials that are based on molecular profiling, so studies such as MOST, for example. What would be really exciting is whether we can adapt the organoid technology to test therapies in real time and provide an answer back to a clinician for an advanced stage patient, whether there's something that could be effective. But that's going to take a lot of work to get that up to scratch, and whether it can ever happen fast enough, I guess, is the issue. So another way to try and obtain evidence is using data that's already out there. And so getting off the ground this MOC FATE, from anecdote to evidence, which is a registry study. And the idea here is to mine all the data that there is internationally and nationally in various registries and compare this current standard of care, so platinum or platinum combination, with other particular chemotherapy classes, or in the recurrent setting, those treated with a targeted therapy, and to see if we can see a difference in time to progression on certain agents over others. And if we can get a hint from existing data, then that might lead us towards perhaps a registry-style trial, which is a little bit easier to set up, and to try and see if we can evaluate that prospectively using data coming in. So with that, I will leave it. I would just like to thank all the lovely people in my lab, the funding agencies, and particularly all the collaborators that worked on the GAMET study and are continuing to work with me. I'd particularly like to give a shout out to AOCS, without which this definitely wouldn't have been possible. The BCFG, Genomics, Core and Bioinformatics at PDMAC. Thanks very much.
Thank you very much, Kylie, for that very interesting talk. Always great to hear your talks, but always sort of makes your heart sink as a medical oncologist. Like, we still give these women carboplatin paclitaxel, as you mentioned, because we don't really know what to do with them. But your data suggests that we could be wasting our time. What do you think we should be giving as first-line treatment?
It's really hard, and I just don't know. Like, you're absolutely right. You have to give them what you have, because the evidence for any other alternative is just not there. And I think that's where I'm hoping that the MOC FATE study, being able to mine that data, particularly more recent data where in other countries where they're, even at the primary stage, women are being offered the choice of being treated with a GI-style therapy, and just to sort of see if there's any kind of signal. But it's really hard.
Yeah, it is really hard. And also a great point that they should ideally be allowed in our studies, but it's actually often a group that's excluded from clinical trials in recurrent ovarian cancer, which is another problem. So we have got a couple of questions from Anna DeFazio. The first one was, are ELF mutations at high variant allele frequency independent of KRAS mutations?
So they're not generally at high variant allele frequency. They're usually, the allele frequency would suggest that they would be heterozygous at most, and some of the tumours, that was actually a fairly low allele frequency, so it could be a subclonal mutation. They were not completely independent of KRAS mutations, but it was statistically significantly fewer KRAS mutations than we would have expected if it had been by chance.
Okay, thank you. And her other question is, given that carboplatin paclitaxel doesn't seem to work in your organoids, have you looked at different types of chemotherapy to see if anything else works any better?
So not systematically. With the very first one, ORG16, we did look at Folfox, and it was resistant to that. That was a particularly nasty tumour, and the patient, Folfox was planned to give her because it was a recurrent case, but unfortunately she passed away before that was possible. So we can't compare to what happened to her to what happened in the lab, other than she had a very aggressive tumour. So that's definitely on the cards for when we have enough organoids. So we've got about 5, I think, carcinoma lines now. I'm just trying to get a few more. It's hard because they're rare, but we'll be starting sort of a more systematic screen through of common chemotherapy agents and their combinations coming up.
And just for the clinicians online with the organoids, it's obviously a newer area. Like, how long does it actually take to grow them to the point where you can do drug testing? And is that feasible within, the disease trajectory for a patient in real life?
So it's quite variable. And there are other tumour types where you can get organoids up and running within a week. And it really depends on how many tumour cells you get in the specimen. So if you get a really good piece that has a high proportion of tumour cells, certainly within a week you'll know that whether it's going to be successful or not. And then within another week or two, you'd probably have enough to be able to do some limited drug screening. But I don't know how easy it's going to be to do for all the tumours that come through the door, for all of the ones that you want to do, which is going to be the high stage recurrent ones. And particularly for the recurrent ones, like I said, the amount of tissue that we often get, because they're not going in for surgery, they're not having a cytoreductive surgery. And so you're sometimes relying on really tiny biopsies, and it's just not enough. I don't think, apart from that very first one, that ORG16, we've had a couple since then from small biopsies, and they haven't been successful in growing. So that would be the limiting factor, is the amount of material that you can get.
Well, thank you very much, Kylie.